Welcome, and thank you for listening to this pre-recorded talk about some of the work of the Duchenne Regulatory Science Consortium, one of the consortia at c-path that's focused on Duchenne muscular dystrophy, which is a rare disease. And I'm really excited to share with you some of the work that we've been doing to try and accelerate drug development and therapy development in general for Duchenne muscular dystrophy. My colleague Varun Agarwal and I will take you through some of the work that we've been doing in developing models to describe Duchenne disease progression and really help us design more effective clinical trials in this disease space. So back in 2015, the first drugs were coming to the FDA for approval, having been through relatively small trials. Several potential therapies have reached phase 3 trials, but no trial had met its primary endpoint in a larger trial. And there was a lot of discussion and argument over the efficacies of these first treatments. You can imagine that treatments go through trials, they don't meet statistical significance. Some people think they work, some people don't think they work, and no one really knows. So this was the environment when we launched the Duchenne Regulatory Science Consortium, next slide, which was a consortium that included industry members, academic members, and Parent Project Muscular Dystrophy, which came together to really try and see what we could do to resolve these issues. How could we get to a point where when we ran clinical trials in Duchenne muscular dystrophy, if the drug worked, at the end of the trial, we'd know for sure it worked. And if it didn't work, we'd know for sure it didn't, and we should move on to something else. And that was what the Duchenne Regulatory Science Consortium set out to resolve. Next slide. So we decided that the way to approach this problem was to build mathematical models of disease progression, which would help us really understand the natural history of disease, how clinical endpoints change with progression of disease, and very importantly, understanding the variability. And we wanted to do this specifically with the goal of trying to understand clinical trials and how to run effective clinical trials. So we wanted to understand the population-wide variability in progression, the causes of variability when possible, understanding if there were subpopulations of patients that changed differently, as well as understanding effects of specific things to trials, such as the dropout, placebo effects, and such like. So we started to build a clinical trial simulation platform based on a series of mathematical models of disease progression as measured by various endpoints, with the goal of being able to optimize clinical trial protocols in the future. Next slide. In order to do this, we needed data, of course. We have now amassed a database of 20 different data sets, all kinds of different data. A lot of it is from placebo-answered trials. There's a strong backbone of clinician-collected natural history data, and then various other types of mainly clinical collected data as well. You can see here, as is typical of a rare disease, some of these data sets were very small. Some of them were considerably larger. They varied in the age range of the patients included. They varied in the length of time data was collected, and they varied in the types of variables collected in each study. What was important here is that although each of the studies had its own unique inclusion criteria and exclusion criteria, they were overlapping. So although some looked only in ambulatory patients and younger patients, some looked primarily in older patients, some studied patients for only a few months. Some, I think the longest we have data collected for is 12 years. And these overlapping studies produced a very strong data set that we could use for analysis to really get at these questions that we wanted to resolve. Click again, please. So out of this, we couldn't use all the data for analysis. There were some data sets that simply didn't have the variables we were interested in, but our total analysis data set is now 1,503 patients with about 25,000 observations in total. Now, obviously, depending which endpoint you're looking at, there may be more or less patients in each analysis. Next slide. In order to bring all these data sets together, we developed a therapeutic area user guide for CDISC standards for Duchenne. And I'm not going to talk about that in detail here. I just wanted to make sure you had the link, because this is a way of standardizing data structure, and is a way that we use to be able to take very different data sets and integrate them together into larger data sets for analysis. And that was published in 2017. So I'm not going to talk about that in great detail. Next slide. But what we were really aiming to do is take the input from all of these different studies and build mixed effects models to really look at the longitudinal change in endpoints, the change in time, and how the various covariates that we know affect Duchenne affected that rate of change. So we were looking at things like age and height and weight, whether the patients were on steroids or not, and their genetic mutations and all sorts of other things that would inform us on the rate at which disease got worse. So we wanted to understand the trajectory of patients, the rate of change, and the predictors of that, because we could use those models to then build a web-based clinical trial simulation platform. Click again, please. So really, this is a summary of what we're trying to do. Is we're trying to take all that data from those 20 different data sets and transform it into knowledge that could be applied to designing new clinical trials and getting information out of those clinical trials about potential new therapies. Next slide. So what are we modeling? Unfortunately, Duchenne is not a disease where we have one endpoint that can be used across the whole spectrum of disease. So we initially started off looking at 6 different potential endpoints. I'm not going to read them off here, but they are essentially a series of different endpoints that measure different functions that are lost at different stages in disease. We ended up dropping the final model of the Brooks scale because it's simply not sensitive enough for use in clinical trials, but we still have models of 5 different endpoints that we're looking at in this population of patients. In addition, we looked at a large number of different potential covariates that might affect that rate of change. And again, I'm not going to read them there. Different endpoints showed sensitivity to different ones of these covariates. Next slide. And click again. So this is an image just really of the data in the database, showing each of those initial 6 endpoints that we were looking at and how they change with age, because of course, Duchenne progresses with age. I'm not going to talk about this in detail. These are not models. This is just examples of the data. Each of those individual circles is an individual observation. And the things I want you to take away from this image is, one, that basically each of those graphs looks like a furball, these huge variants in the population. And that was what we were trying to understand, is can we limit or understand some of that variability so that we could have patients enter into a trial that would know how they would progress over time. The second thing to take away from this is none of these endpoints change in a linear fashion. In all cases, they increase initially with the growth and development of the child, and then they decrease due to the disease. So we couldn't just create linear models. These were going to be complicated, non-linear models that needed to be developed to explain the change over time in these endpoints. So that was the puzzle that we gave ourselves to do, is could we understand the progression of these patients, understand the variance in these patients, and really understand how these endpoints could be used in clinical trials. Next slide. So being the lovely person I am, I handed this over to our quantitative medicine team and asked them to solve the problem. And with that, I will pass over to Varun from our quantitative medicine team, who's been doing much of this work. Varun.
Thanks, Jane. Thanks for providing this background to the problem. And hello, everyone. My name is Varun Agarwal. I'm a scientific director in QuantMed at c-path. So today I'm going to talk about how we develop these disease progression models. And before I get into that, I just need to talk about what the overall workflow is. So the first thing is basically looking at these 5 endpoints, and the problem at hand is that we have to describe the disease progression of DMD over the years of age as it is measured by these 5 endpoints, and these being the FVC, NSAA, velocity climb, velocity stand, and velocity walk and run. The first thing that you do is you identify a base model, and this is the first step in any disease progression model, that you have to identify an accurate base model. And as I will show in subsequent slides, how that was done. So once we identify a proper base model, different kind of diagnostics are performed, a popular one being the visual predictive check. And that will be shown in later slides as well. The next thing is to incorporate covariates, and this is done in order to explain the between individual variability. And once the covariate model is properly identified, it is validated as well. And again, there are different ways of validating the model. What we did was an external validation, and an example of that would be shown in the later slides as well. And overall, you do them in a sequence. So the first you do is you identify a base model, then overall you move on to a covariate model, and finally you have an understanding of the trajectory of a disease, in our case, DMD. and over a period of specific age, and we do understand what their predictors are as well. So jumping back to the very first step, which was identification of base model, and as Jane alluded to, DMD is a progressive disease in which muscle loses their function over the course of disease. However, people with DMD do exhibit an increase in the muscle function, primarily due to growth in the earlier years of the disease. So we need a base model which describes both the increase and decline phase. So if you look at, as an example, and this is, again, it's not a simulation, it's just an example plot or a representative plot. So here I'm showing forced vital capacity or FVC as it changes over with the age of a patient. So typically what you would observe is that you would see that FVC is increasing, and then as the disease becomes more and more progressed, you start seeing a decrease in the FVC. So essentially, to model this, we have to include two models. One which we call a Chapman-Richardson growth function, which describes the growth phase, and then the decay phase is described by the sigmoidal decay function. And both of these models are typically used in pharmacometrics and other fields. Chapman-Richardson growth function is very popular, and an example where it's used is in the forest, where people want to estimate how trees would grow. So for the math nerds like me, this is the equation, and I don't want to go into too much of detail, but this one on the left side is the Chapman growth equation, and this is, you see that it would saturate to G max, and on the right side is the sigmoidal decay. Different parameters have different meaning and different significance, which you can read here, but the two which I want to point out is, one is gamma, which is referred to as the Hale coefficient or shape factor. Higher the value of gamma, the steeper the TK. The next thing was to look at what different covariates are there which describe or predict the progression of the disease. So we looked at several. Among them were baseline age, were the patients on steroid at baseline, what kind of study it was, what the baseline score was, the race, and the specific mutation. And again, these covariates are highly data dependent. So if you have richer data, we can probably look at the covariates in more detail, but with the data that we had at hand, these were the only covariates which we could explore. And as you can see here, here we are plotting the variability, which is described by ATA-DP50 on the y-axis, with baseline score on the x-axis, and as we stratify it with age, and again, the legend is shown at the bottom, you do see that if you specifically look at these green dots here, you do see the trend line, again shown in green solid, there is a specific trend. So these covariate models were incorporated based on different mathematical models, mathematical functions, and owing to time, I will not go into detail how that was done here. But finally, all of that came together into a final model, and an example of that here would be FVC. So for forced vital capacity, we had baseline score and baseline age as a continuous covariate added using the power law on DP50, and the race and the mutation were added on Gmax and DP50 as categorical covariates. And the result of that was that 60% of variability on Gmax and 30% of the variability on DP50 was explained by covariate. The next thing to do is to look at, is this model even making sense? How valid this model is? So to do that, we looked at an external data set and did something called a visual predictive check, which I think is on the next slide. But here are typical diagnostics, which is shown here. So if you look at this plot on the on the top right, you see I have plotted observed force vital capacity with predicted force vital capacity, and then this is randomly distributed around the 45-degree line, which gives us a good confidence in our model, showing that our model fits the data well. And these are the other kind of goodness of fit indicators. And as I was saying, the next thing was to look at was look at an external. This keeps skipping. The next thing, as I said, was to look at the visual predictive check. So this is that. So to build this plot, we looked at an external data set. The individual observations of those are shown in these black dots, and then ran 1,000 simulations, and the results of those simulations are shown in these bands. Essentially, what this is showing here is the median band, which is shown in blue, and the blue solid line is actually the median. The band is the 95% confidence interval around that median. The greens are respectively the 5% and the 95% 95 percentile. And then this is the bands are again the 95% confidence interval around those lines. So typically what you look at in this kind of a VPC is that the data which is shown by this dashed line, is that lying inside the band or not? And in our case, it does. So for us, this was good enough validation, considering that this data set was external and was not used at all in developing the model. So the question then is, why do this? What will these models do to help? So the first thing is that if you develop a mathematical model, we develop a mathematical understanding of how these endpoints change over the disease progression. We also understand what the sources of variability is, and then we can put together all this information in doing simulations to test different clinical trial designs, so as to have an optimized clinical development. And then all of these could be used to predict significant clinical endpoints, clinical outcomes rather. For example, time to loss of ambulation, respirator use, etc. And this is what a typical clinical trial simulation tool looks like, and it's a work in progress. So as you show in this window, see in this window here, we can modify the clinical design characteristics by changing the number of subjects, the follow-up assessment intervals, and we can look at the baseline patient features. We can look at assumed drug effects, and then finally, once we do all of this, we can have a simulation which is chosen by the user in the sense at what they want to look at. So this could be used by pharmaceutical companies or sponsors to optimize and design their trials. And then with that, I think I'll pass it on to you, Jane.
Thank you. So just to conclude, all of that work that Varun has just described, and this was work that was done by our collaborators at the University of Florida, Sarah Kim and Karthik Lingamani. Our internal team, Varun himself, has put a lot of time and work into this, as has Rhoda Muse and Jackson Burton and Nathan Hanan. The idea of all of this is to really help us describe the disease progression in patients from about age 5 until end-stage disease in terms of the dynamic of change of these outcome measures that we can use as endpoints in trials, and understand how those outcomes relate to each other and what covariates affect the rate of progression. Why do we want to do that? So we can choose optimized populations in a clinical trial, so we get results that give us definitive answers as to whether a drug works or not in a reasonable period of time, using as few patients as possible. Across all rare diseases, we have a challenge in not having many patients. That means we don't have a whole lot of data points to build models like this. And here we've shown that by bringing together data that already exists in the ecosystem, even for a rare disease, we could put together a data set large enough to do some of the sophisticated analysis that will really help drive drug development forward. It will drive investigator confidence in setting up a clinical trial in this disease, because they'll understand what to measure, who to measure it in, and how long that trial is going to take. And that's hugely important to companies developing drugs. So what we've aimed to do here was really provide tools to help others develop drugs for this very rare and deadly disease. These models aren't quite complete, but we are really close now. The next steps will be to finish those final touches to the models and the clinical trial simulation tools. And we are going through both FDA and EMA pathways to seek endorsement of the models to make sure people can use them with confidence. And they understand that the regulators have already reviewed all of this complicated maths, and they are okay to go ahead and use these in designing their next clinical trials. Next slide. And we hope that really means that in the future, when we do clinical trials in Duchenne, we'll really know if a drug works or not. And if it works, great, we can move forward. And if it doesn't work, we can get rid of the drug and move on to something that perhaps will. Next slide. So finally, I just want to thank everybody who's done this work. I mentioned that the actual modeling work was largely done by the University of Florida and our QuantMed team in collaboration with our data center, who put all the data together. But it's also the intellectual work of the entire D-RSC consortium. We've had at various times between 6 and 8 companies involved in the consortium, about 20 academics, liaisons, advisors, and all sorts of other people working towards this. You can see more about our work at our website that's there. You can send questions to me at the email address that's there. But thank you very much for listening. We're excited about this work. And as we build out IDC ADAPT, we hope to be able to see this and do similar projects across many other rare diseases. Thank you.